Good evening, everyone. Welcome you all to the NPTEL TA session of Week 8 lecture series. Today we'll be talking about some assignments and questions on applications of genome editing in treating human diseases. So before starting, let's wait for a few minutes. Let other people join, then we can start. Okay, let's start then. Uh, this is the first question of today's assignment. So, a patient is diagnosed with absent or reduced production of both delta and beta globin chains. Which type of thalassemia does he have? If it is delta thalassemia, beta thalassemia, delta beta, or gamma thalassemia, which one it will be? Please try to answer this question in 2 minutes, then we will discuss it. Okay, let's discuss it. So look at the picture here. So what i- what is the disease we are talking about? Thalassemia. Okay. So here, these are the normal red blood cells. Here you can see white blood cells and red blood cell, healthy red blood cell and platelets also in the blood. But in case of a thalassemia patient, you can see here the malformed red blood cells or RBC. Okay, the structure of RBC of a normal person, it is spherical, but in case of thalassemia patient, the shape changes. Okay, and there are other blood components, but the thalassemia doesn't affect white blood cells or platelets. It only affects the RBCs. So it is a hereditary disorder and it is inherited by autosomal recessive manner. Okay, it arises due to defects in hemoglobin production, and it is different from other hemoglobinopathies like sickle cell anemia, where the RBC becomes in the shape of sickle, and it is caused by structural defects in hemoglobin. Okay, so whereas thalassemia is caused by decreased synthesis of alpha or beta chains of hemoglobin. So, how does thalassemia occur in a person? Generally, it transfers from parents to offspring via autosomal recessive manner, or and it is generally caused by decreased synthesis of alpha or beta chains of hemoglobin. Fine, and depending on the type of polypeptide chain, that is, if it is alpha chain or beta chain whose synthesis has been impaired, the type of thalassemia has been classified. Okay, so they can be mainly categorized as alpha thalassemia, that is absent or decreased production of alpha globin chains. Next is beta thalassemia, which is absent or reduced production of beta globin chains. Next is delta beta, which was our question, that is absent or reduced production of both delta and beta globin chains. So now you know the three types of thalassemia which has been classified depending upon the polypeptide chain synthesis. Okay. Next is rarely occurring thalassemia, which has less significance. That is delta thalassemia. It is absent or decreased production of delta globin chains, and gamma thalassemia. It shows decreased production of gamma globin chains. So nothing to be confused about the type of the thalassemia disease or the classification, because if there is decreased production of alpha globin chain, then it will be called as alpha thalassemia, and if there is decreased or reduced absent or reduced production of beta globin chains, then it is called beta thalassemia. So the answer of our question is delta beta thalassemia. Fine. Let's move to our next question. How many mutations have been identified so far worldwide known to cause beta thalassemia? If it is 1050, 105, 350 or 3050. Please try to answer this question in 2 minutes, then we will be discussing it. Okay, let's discuss then. So, in a normal human diploid cell, there are two copies of each chromosome, and two copies of beta globin gene are present, as one copy is present in chromosome 11. Okay. If one copy of beta globin gene becomes impaired, mild anemia may arise. If both copies of the gene become impaired, severe anemia and potentially life-threatening condition arises. Beta thalassemias are more majorly caused by non-deletions type mutation, including single nucleotide substitution and short insertion or deletion leading to frameshift mutation. And this deletion in beta globin gene upstream locus control region, that is LCR, are rarely seen in beta thalassemia, and there are about 350 disease causing mutations have been identified so far worldwide. Okay, so here is our answer is 350. Okay, so we have discussed about beta thalassemia here. How beta thalassemia happens? It generally happens due to the reduction of or absence of beta globin chain of hemoglobin. So, and it generally starts from the chromosome 11, which has two beta globin genes in a pair. If one pair becomes impaired, then mild anemia arises, and if both of the gene becomes impaired, then it leads to severe anemia. And there are about 350 disease causing mutations have been identified worldwide. Okay, so let's see how this chain works. Okay, so it can be absence of beta globin chain or reduction of beta globin chains. So which leads to excess of alpha globin chains. Fine. That leads to two types of consequences. One is absence of beta globin chain, which leads to precipitation in erythroid precursors within the bone marrow. Next, it leads to ineffective erythropoiesis. Okay. Then, if which leads to anemia, and if there is reduction of beta globin chain, that leads to membrane damage to peripheral RBCs, which leads to hemolysis, and that ultimately leads to anemia. And anemia leads to three types of consequences. It can be erythropoietin increased, absorption of iron is increased, and blood transfusion. So, if erythropoietin is increased, it leads to expansion of bone marrow, and skeletal changes happens, which can be called as bone deformities. Fine. Or if absorption of iron increases, then that leads to iron overloading, which leads to cardiac death. And blood transfusion, if you require blood transfusion, that also leads to iron overloading, and which ultimately leads to cardiac death. Fine. So this is the whole uh, chart or flow chart of beta thalassemia, how it happens in a normal, how it differs from a normal human being to patient, beta thalassemic patient. Okay, so let's move to our next question, which is which type of alpha thalassemia is characterized by mild to moderate mental retardation? If it is ATR 16, ATR 26 syndrome, HB Barth syndrome, or ATR 116 syndrome. ATR stands for Alpha Thalassemia Intellectual Disability Syndrome. Which one it will be? Please try to answer this question in 2 minutes. Then I will be discussing. Okay, let's discuss then. Alpha thalassemia mental retardation X linked, that is ATRX. So here in the figure you can see that erythroblast, if it has excess of beta chains, that leads to alpha thalassemia. That is mutation of gene that encodes for the alpha chain located on chromosome 16. That is due to mainly deletions which lead to alpha thalassemia, and these are the inclusion bodies. Fine. Next, if there are excess of alpha chains, that leads to beta thalassemia, and mutation of gene that encodes for beta chain located on chromosome 11. So, mutation of gene for beta chain located on chromosome 11, and mutation for alpha chain is located on chromosome 16. So, and that mutation in the beta chain, which leads to excess of alpha chains, is called beta thalassemia. Fine. So there are two types of thalassemia. Now we have discussed it completely, and I guess everyone is clear. So a type of alpha thalassemia known as ATR16, that is alpha thalassemia intellectual disability syndrome, is also characterized by mild to moderate mental retardation. And this ATR16 syndrome is caused by a deletion of part of chromosome 16. So if we are talking about alpha thalassemia, then it is related to chromosome 16, and if we are talking about beta thalassemia, then it is related to chromosome 11. A band on the short end of chromosome to the end of the chromosome. Fine. So your answer will be ATR16, which leads to moderate, mild to moderate mental retardation. You can go through the whole lecture notes or lecture series, your week 8 lectures to uh, understand it more about the ATRX and other thalassemia related condition. Okay. Let's move to our next question. Which of the following risk is not associated with blood transfusion therapy? If it is alloimmunization, blood-borne infection, severe allergic reaction, or compromised immunity, which one it will be? Okay, let's discuss then. So, blood transfusion dependent, there are two types of patients, which some of them require blood transfusion from time to time, others don't require it. So those who need blood transfusion, they have some complication to face. That is kind of reduction iron toxicity on vital organs, which can damage your liver or kidney or heart because of excess iron intake to the body. Okay, so let's discuss about it more. Transfusion of RBC is done in transfusion dependent patients to maintain adequate hemoglobin level. Thalassemia major or intermediate patients require blood transfusion at regular intervals. There is risk associated with transfusion therapy as it causes alloimmunization and blood-borne infection. Okay, if the person providing blood and the packet of blood which is transferred to the thalassemia patient, if it has some kind of infection, then that can transfer to the patient also, and that all and the other immunization problems, that is alloimmunization, is kind of more complicated problem for the beta thalassemia patients. However, advancements made in blood product screening and preparation has aided in this. Iron overload or hemosiderosis due to ineffective erythropoiesis is another concern of blood transfusion therapy, which leads to liver fibrosis and other complication. Okay, so ineffective erythropoiesis, uh, and that leads to iron overload, and which and for the consequences it leads to liver fibrosis and other complications. Okay, so here the question is, which of the following risk is not associated with blood transfusion therapy? So not associated means alloimmunization is associated with blood transfusion therapy risk. Next is blood-borne infection. Severe allergic reaction can also happen. Compromised immunity can be one risk which is not connected to the blood transfusion therapy. Next question is, which of the following is not true about severe combined immunodeficiency, SCID? A group of rare disorders caused by mutations in different genes. Mutated genes involve development and function of immune cells. X-linked SCID primarily affects female infants, or SCID is inherited in an autosomal recessive pattern. Which one it will be? Okay, let's discuss it. Severe combined immunodeficiency diseases, SCIDs, are a group of rare and life-threatening diseases caused by monogenic defect. Okay, it is one of the most severe forms of primary immunodeficiency disorders, and it is characterized by defect in number and function of T cells, which also affects the B cells and NK cell population. NK cell means natural killer cells and B cell population. The incidence of SCID varies between 1 in 40,000 to 75,000 per live birth. Okay. So this is the pedigree analysis of SCIDs, where you can see here it is transformed in a autosomal recessive manner and also in a X-linked recessive manner. Okay, this all form of SCIDs are autosomal recessive and X-linked recessive monogenic disorder, except SCID types, disorders anomaly, and some cases of the autosomal dominant syndrome. Fine. So here you can see here the male is a carrier and female is also a carrier parent. So that leads to affected female child, while the another female is carrier and unaffected child and carrier child. So that means out of four child, three offsprings has been affected. Okay. So in case of X-linked recessive, it also affects the male. If the mother is carrier, then it affects the male child, and if is the father is carrier, then it affects the female child, that is their daughter, but it is in carrier format, not completely affected. Okay, but if the mother is the carrier, then it can affect the male person of the out of 1 is to 4 is SEO, and it is a diseased patient. Okay, so this is the pedigree analysis of SCIDs in two types, that is autosomal recessive manner and X-linked recessive manner. Fine. Then let's move to our next question then. What percent of SCID in children are caused by reticular dysgenesis, that is RD? If it is 5%, 50%, 4%, or 2%? Okay, let's discuss then. Classification of SCID is based on underlying genetics and prevalent molecular pathogenic mechanisms that can be defective survival of hematopoietic lineage precursors, and VD is a recombination and alternations in TCR, cytokine signaling abnormalities, toxic metabolite accumulation, TCR abnormalities, that is T cell receptors, or thymic abnormalities. Fine, they are categorized into few terms, and we are talking about defective survival of hematopoietic lineage precursors. Adenine kinases are responsible for cellular and mitochondrial energy homeostasis. And this adenine kinase catalyzes the reversible transfer of a phosphoryl group from adenosine triphosphate ATP to adenosine monophosphate AMP. And this ATP, ADP, AMP concentrations maintained within tightly regulated range is crucial for cell survival. And around 2% of children with SCID are caused by RD. Okay, so here is your answer. That is, what percent of SCID in children are caused by reticular dysgenesis? That is RD. So your answer is 2%. Okay. So next question is, lower levels of which clotting factor cause hemophilia? Either factor 8 or factor 9, factor 12 and 11, only factor 10, none of the above. Earlier we have been talking about the disease thalassemia. Now we need to talk about hemophilia. Okay. And we also talked about SCIDs. So genome editing and engineering is a powerful tool which has been helping in curing several types of diseases. But here we have been talking about some examples, including some rare conditions like SCID and thalassemia and hemophilia. Okay, so please try to answer this question, then we will be discussing it in a few minutes. Okay, then let's discuss. So this is the pictorial representation of the difference of RBC or blood vessels showing uh, between healthy person and hemophilia person. So it is the normal blood vessel. Okay. Now injured blood vessel clotting factors are activated in the second step. In case of hemophilia patient also, injured blood vessel clotting factors are activated. But in the third step, a healthy person will find that platelet block formation happens. But in case of hemophilia patient, lack of clotting factor happens, which leads to weak platelet block formation. Okay, which is the as a consequence it leads to step 4, where incomplete or delayed fibrin clot happens. That means when the blood vessel is injured and blood starts flowing outside the cells, then it becomes uncontrollable for a hemophilia patient. But a healthy patient, there is the fibrin clot and which is stable, so that it is the blood vessel can be repaired within few time. Okay, but in case of hemophilia patient, if blood starts flowing, then it takes a lot of time for clotting. This is the visual difference between healthy patient and healthy person and hemophilia patient. Okay, so there are few factors which is responsible for our hemophilia. That is, it can be classified into hemophilia A, that is factor 8 deficiency, hemophilia B, factor 9 deficiency, hemophilia C, factor 11 deficiency. Okay, so there are three factors which is responsible for hemophilia, that is 8, 9 and 11. So here is your question is, lower levels of which clotting factor cause hemophilia? So it can be 8 or 9. Okay, but here it is written 12, so it cannot be considered for hemophilia, and 10 is not there, and none of the above, let it be. So either factor 8 or 9 can be a cause of hemophilia. Next question is, which of the following pair on type of hemophilia and its deficiency factor is correct? So I just discussed it in a few minutes ago. Please try to recall your memory and answer this question. Hemophilia A, factor 9 deficiency. Hemophilia B, factor 9 deficiency. Hemophilia C, factor 9 deficiency, or hemophilia B, factor 11 deficiency. Which one it will be? Okay, then let's discuss. So we have just discussed that factor hemophilia A leads occurs due to factor 8 deficiency, hemophilia B occurs due to factor 9 deficiency. So your answer will be B. Okay. And hemophilia C occurs due to factor 11 deficiency. Hemophilia A, also called classic hemophilia, is the second most common type of all hereditary bleeding disorder after VWD, and the most common type of hemophilia in the world. Hemophilia A is 5 times more common than hemophilia B, and hemophilia A and B are the only HPDs that are inherited in a sex-linked model, and the affected gene is located on long arm of X chromosome. Okay. Hemophilia C, it is the rare form of hemophilia. It is an autosomal retreating disorder, and bleeding symptoms due to the deficiency of factor 11 are seen in hemophilia C. Okay. So here is our next question. Which of the following can be caused by an intrachromosomal inversion involving intron 22 of the coagulation factor 8 gene? If it is mild hemophilia B, mild hemophilia A, severe hemophilia B, or severe hemophilia A? Which one it will be? Okay, then let's discuss. Half of the cases of severe hemophilia A are caused due to intrachromosomal inversion involving intron 22 of F8 gene. Okay, so your in this is your answer, severe hemophilia A. And I guess we have discussed it just a few minutes ago. This is the difference between healthy person and the person with hemophilia, and how the genes work and the factors work. Okay. Next question is, which of the following characterize the TB plus severe combined immunodeficiency disease, SCID? Please read the options carefully and try to answer. If it is defect in both T and B cells, or the absence of mature T and NK lymphocytes while B cells are present in increased number, or the absence of B cells which while mature T and NK lymphocytes are present in increased number, and or absent or decreased production of B cells. Which one it will be? Okay, then let's discuss. So TB plus SCID, it is characterized by the absence of mature T and NK lymphocyte, mature T cells and natural killer lymphocytes, while B cells are present in the increased number. Okay, and this X-linked recessive SCID include XSCID, and that arises due to mutations in IL-2RG gene, that is very low T cells, normal to high B cells with low immunoglobulin levels. And this autosomal recessive inheritance of TB plus SCID includes JAK3 deficiency, IL-7R alpha deficiency, CD45 deficiency, CD3 delta deficiency, and others. Okay, so this TB plus SCID, what is it? What it is? It is characterized by the absence of mature T lymphocytes and mature natural killer cells, while B cells are present in increased number. Okay, so I think the name suggests the definition. So we need to understand the meaning and try to remember it. Okay, so here is your answer. Okay, the absence of mature T and NK lymphocytes, while B cells are present in increased number. Okay, and here is the flow diagram of TB plus or in TB minus. Okay, severe combined immunodeficiency, and it while it happens due to defective humoral and cellular immune system. This can be TB NK minus or TB NK plus. Just a minute. TB NK minus, which can be of two types, that is reticular dysgenesis. We have talked about it recently, RD. About 2% of children has been affected with RB, that is AK2, and ADA, which also leads to uh, conditional dysphasia. Next is TB NK plus. This can be microcephaly plus or microcephaly plus growth retardation, or without microcephaly. It can be divided into two types depending upon the microcephaly family. TB plus, it is also divided into two types, that is TB plus NK minus and TB plus NK plus. Where it is represented in the chart format, we need to go for each and every detail of CD cells, then we can understand it. Okay, I will highly suggest you to go for your lecture notes and lecture videos where you can understand it in a better way. So the next question is, in the first trials of gene therapy for SCID, retroviral vectors were used in which expression of the normal transgene was driven by the retrovirus long terminal repeat LTR, self-inactivating retroviral SINRV, graft versus host disease GVHD, or immunoglobulin replacement therapy IGRT. Which one it is? Please try to answer this question. Then we will be discussing it in a few minutes. Okay, let's discuss it then. So it shows the schematic representation of gene therapy protocol used in SCID-X1 clinical trials, where CD34 plus hematopoietic stem cells are sorted from bone marrow or mobilized peripheral blood stem cells using magnetic separation and cultured with cytokines. And these cells are transduced with a specific vector containing the corrective human interleukin 2 receptor G gene, that is IL2RG, and after a period of culture the cells are reinfused in the patients. In most recent clinical trials, the cells are infused after a low dose conditioning treatment. Subsequently, the immune reconstitution starts and the patient is theoretically cured for life. Okay. So, in the right side, it shows a schematic representation of how the patient is cured. That is HSPs, that means hematopoietic stem cells are obtained from the patient's bone marrow or mobilized PBSCs, that is peripheral blood stem cells, which contain CD34 plus cells are sorted and harvested and activated. Okay. Next, CD34 plus cells are transduced with the vectors containing corrected copy of IL2RG transgene. Next, gene-corrected CD34 plus cells are reintroduced in the patient with or without conditioning. So now the patient is cured because only the CD34 plus cells having defect has been sorted and harvested. Then the defect has been cured with using the vectors, and that is inserted into the patient. Okay, this generally happens in the during the children age. In the first trials of gene therapy for SCID, retroviral vectors were used in which expression of the normal transgene was driven by the retrovirus long terminal repeat LTR. Okay, so here is the question and the answer is LTR. Okay, retrovirus long terminal repeat. With this approach, successful and durable T cell reconstitution was achieved in patients with XSCID and ADA-SCID. Unfortunately, several patients developed leukemia due to preferentially integration of retroviral vectors in proximity of transcription initiation sites of genes and by the strong enhancer activity of the viral LTR, leading to increased and deregulated expression of the targeted oncogenes. Okay, to overcome the above problem, self-inactivating retroviral SINRV vector in which the U3 enhancer was deleted from the LTR and expression was driven by the weaker eukaryotic human elongation factor 1 alpha SORT promoter was developed as safe alternative. And additionally, SIN lentiviral vectors have been developed to reduce integration within proto-oncogenes. Okay. So this was all about the gene therapy for SCID. Next question is on natural killer cells. So the question is, natural killer cells are the predominant innate lymphocyte subsets that mediate anti-human or antibacterial responses, anti-tumor and antifungal responses, antihistamine and antiviral responses, or anti-tumor and antiviral responses. Which one it is? Please try to answer this question. Okay, let's discuss. So this is the 3D format of natural killer cells, 3D image. Natural killer cells are the predominant innate lymphocyte subsets that mediate anti-tumor and antiviral responses. So here is your answer: anti-tumor and antiviral responses. NK cells recognize and kill cancer or virus infected cells and therefore possesses promising clinical utilization. NK cells account for 5 to 20% of the mononuclear cells of the peripheral blood and the spleen. They produce cytokines, among which interferon gamma delivers signals to the innate component of the immune system, which activate the inflammatory process in defense of an organism. Okay, so this is all about the function of natural killer cells. So, okay, this was the last question of today's session. Thank you for joining. Thank you all. Uh, Please try to go through your lecture videos and lecture notes. There you will find in very detail of each and every treatment using genetic engineering of different types of diseases. Specifically, here we have talked about hemophilia, SCID, and thalassemia. So, the lecture notes and lecture videos contain the details of each and every disease, but here we have considered some questions of few parts only. So I hope everyone is clear with all the questions. If you have any doubt, then you can ask me. Do you have any question? Is there any doubt you want to ask? Okay, then see you in next session. Thank you. Thank you for joining.